Okay.
Right. Hi, my name is Benjamin Lewis, and today I'll be talking through a research proposal for my senior research that I'm actually doing right now with Dr. Woodward, who is a biochemist and organic chemist here at the University of Mount Union. And this research will be taking place starting last year up until the spring of my senior year in 2017. So what we're really looking at today is how bacillophile, which I'll explain later, actually inactivates fossamycin, an antibiotic, because the antibiotic resistance, which we kind of briefly touched on in class. But before we do that, I really want to talk about why this is a major issue. So if we look here from the Center of Disease Control and Prevention, or the CDC, as years increased, even from 1981 to 2001, and progressing even onward in present day, if we pay attention to MRSA, which stands for multi-drug resistant Staphylococcus aureus, which is a bacteria, they're becoming more and more resistant to a lot of our common day antibiotics. This is in the news very prevalently, but one of the issues that we don't discuss a lot is, as the World Health Organization points out, as these bacteria are becoming more and more antibiotic, the companies researching new and improved antibiotics is actually decreasing now to what was in 2011 around 4 different companies now. So one of the reasons that this antibiotic resistance is arising is, you might have noticed at your doctor's office, some of these doctors and physician's assistants and nurse practitioners are handing out antibiotics left and right. Why? Well, part of the reason is your own mother's fault, because they go in and their son or daughter has a cold and something that's a virus, and they beg the doctor for some sort of medicine. And doctors sometimes will appease to that and give them antibiotics even when they're not necessary. Another problem we face is that full antibiotic dosage are not actually completed. So when we're sick and we have bacterial infections, we're starting our antibiotic regimens, but not finishing them. What that means is that we're not actually killing every single last bacteria in our body, and the ones that survive maybe have susceptibility and ability to start to become resistant to the bacteria. And finally, one of the other present issues is flushing these antibiotics down the drain, or disposing of them improperly. And what happens there is when you dispose your antibiotics down the toilet, what you may not have done, but it happens pretty frequently, is they become very diluted. So when they're going into our main water systems, you have residual bacteria that are there along with our antibiotics in non-lethal concentrations. And what that means is our bacteria will be exposed to our antibiotics in a way that won't kill them. So when they're exposed to these antibiotics, they're able to start to build up those mechanisms to fight up against it. One of these bacteria that we've had a problem with, is what I was talking about, is that Staphylococcus aureus. We'll talk about this more in lecture from what I'm presuming from future notes, but what we have discussed is the difference between gram-positive and negative bacteria. Well, Staphylococcus aureus is a cocci or circular-shaped bacteria that is a gram-positive. And what that means is, what Shively is telling us here, is that our gram-positive bacteria have a much thicker cell wall made of peptidoglycan, which your lab instructor and your lecture instructor may have talked about. Well, one way that we can treat and kill bacteria that are gram-positive that have those thick cell walls is with fossamycin. This figure up here by Castagnati and Garcia in 2013 shows the structure of fossamycin. Fossamycin works by going in and actually stopping the synthesis of cell wall production. So if these bacteria are no longer able to make their cell walls, they will actually die, and we can treat and kill our bacteria in our body that way. But something that we really need to pay attention to, which maybe you haven't had organic chemistry yet, is this epoxide ring which I have structured up here. And what that is it's kind of 2 carbons with an oxygen in the middle. And it's somewhat of a reactive group that makes this very vulnerable to antibiotic resistance, which we'll talk through later. But when we look at this by Thompson in 2014, he's one of the guys who's really proposed a lot of talk about what bacillophile does. And what we see is that fossamycin, when it goes into the body, is actually attacked, kind of beat up in a organic chemistry reaction where bacillophile, which I'll show you the picture of next, is able to come in and open up the fossamycin epoxide ring. So as we see here in this figure by Thompson, the fossamycin here on the left, and your left, is much different in structure than the fossamycin on the right. So if you look on the far right, you see that BS group, that stands for bacillophile, that's coming in and opening up that ring. So if we look back at our themes that unify living organisms, structure dictates function. Alright, so if we change the structure of our antibiotic, we change its function. And as we change the function and structure of fossamycin, it's no longer able to inhibit cell wall synthesis. So it's no longer effective. So walking through this, if we see here up on the left, we see bacillophile on top. And just to kind of show what's happening here in a more illustrative way is that bacillophiles coming in is attacking that epoxide group, which I was talking about here on this fossamycin, to give us that ring opening. Another tidbit of information which may not be important, but if you're interested, what actually coordinates these two groups is called FOSB, an enzyme within the body, but that's for something later on we can talk about. So what I want to talk about next is where this research is going. So bacillophile, that structure I showed you in the previous slide, is actually made up inside of the bacteria within the use of 3 different enzymes. There's 3 different tools that kind of go along in stages to make bacillophile. Well, our research is actually to block the second enzyme within the pathway. So if we block that second enzyme through the use of our own molecule that we're making, we can block the production of bacillophile. Well, that's awesome because if you get rid of bacillophile, fossamycin can work again. So our research is to actually make this big, gigantic, complicated molecule as shown on the slide here. And what I want to do next is kind of walk through simplistically the stages and steps to do that. So just to reiterate though, our molecule will come in, block that second enzyme, and block the production of bacillophile. You're going to see this slide a lot, but kind of remember it because this is the take-home message, is to block that second enzyme to stop the production. So, how do we do it? Well, that's a great question. So in the first step, what we do is we take a starting sugar. It's a very simple sugar that we take, and we treat it with different organic compounds to actually remove this acetyl protecting group. And what that does is it makes it more reactive by adding that oxygen and hydrogen group on the right side that you see. And now that we have that oxygen and hydrogen group, what we can do is add this chlorocyanide group, which actually comes in and attacks that hydrogen to give us this group. You'll all learn about this a little bit later, but I just want to kind of walk through the simple steps of what's happening. So later on, you'll be like, oh, that's pretty cool. So the next step is actually to go through and add this next big complicated molecule onto where that previous group was. These were taken from a different one, but the major synthetic person we're following in this procedure is Sharma, who developed this protocol in 2011. So now what we need to do is once we have this big complicated group on that part of the molecule, well, we need to add our second major group onto that middle carbon, as you see here. There's that nitrogen-hydrogen group that we need to add. So we're taking that regular nitrogen, that N3 group, and turning it into an NH2 by adding some other reagents. I know this may not be interesting, but it's important stuff, so bear with me because this is the last step. And what we're going to do is finally take that NH2 group, and we're going to add this big complicated molecule. This is shown by Liu et al. This is a different group that was kind of going through this slide in this protocol, but this is brand new research to add this final group onto that carbon. So now that you kind of get the basic steps of how to make it, and you've seen the synthesis, we'll get to the fun part. So now that we have our molecule, we can actually propose our trial and see if this is actually going to work. So what we're going to do is have 2 different agar plates. One's going to have, both of them will have our Staphylococcus aureus, that gram-positive bacteria that I showed you previously in this slides. And one of them will be held as control, and one of them will be our experimental. And we know under normal conditions, our fossamycin under our control group is going to be pretty resistant to fossamycin. We don't expect a whole lot of cell death there. But on the left, what we can do is treat our molecule and give that to Staphylococcus aureus with our fossamycin. And to go back and repeat these slides one more time, what we expect is for our molecule to block the second step, to block bacillophile. When we don't have bacillophile, we don't get the change in structure of fossamycin. So now we just have fossamycin now. They can actually have the same structure and go in and kill the Staphylococcus aureus and hopefully solve the resistance of antibiotic resistance. Thank you. Here are my references. And are there any questions?